Hello everyone. We're going to discuss today how can we consider the microbes which we think as harmful as a resource. Microorganisms are those organisms, living organisms, that we can see with our naked eye, like the bacteria, like fungi, like the algae, like ones over here is called the Archaea, one of the oldest living microorganisms on the planet, also protists, it means the algae. But there is another information we must consider, which is the viruses and the prions are not classified as living organisms. But the question comes that why we say the microbes and relate them as a harmful thing. Yes, if some of the food is accumulated over your tooth, you are going to be harmed because they are going to feed on that. But on the other way, we must not forget that each day the yogurt you eat, the yogurt which is made is mainly from the microorganisms, is cultivated to come with it. So it's harmful if you leave it, it's beneficial if you can use it. Also, we can't miss the role of them in nature, because carbon dioxide in the atmosphere is not only fixed by the plants, but also it's ruled through the earth, it's ruled through the ocean and through the water and comes back to the atmosphere. All of that cycle controlled by the animals and microorganisms, but mainly the microorganisms. Okay, on the other hand, the nitrogen cycle in the atmosphere and the land and through the living organisms like the humans or the animals, all of those nitrous, nitrite, all of those chemical reactions, as we can see them over here, are carried on by microorganisms. And each one of those reactions, we can see the name of the distinguished microorganism that is carrying it on. Sulfur, same story. That's why we must study bacteria. That's why I prefer to make this presentation, because they are the oldest in Earth. Nothing works without them. Basically, and most studied organisms in all the labs, they are found everywhere. And as we have seen, you can find them in your teeth, you can find them even in your skin, and in the most harsh conditions on Earth, on bones, or something like that. Well, if we are going to discuss the bacteria, so how they are mostly studied? They are mostly studied because of the simplicity. A is representing the bacterium, B is representing our cell, the eukaryotes, the higher living organisms. So the prokaryote represents the simpler organisms like the bacteria, like the fungi. The eukaryote represents the animals and plants, and of course the humans. If we can see the simplicity between those two, we can see and figure how much it's simple to use the bacterium in research, because it's just simple to use the genetic material. Here is the DNA, and there are the outputs of the DNA during transcription and translation and the growth of the amino acids. All of that comes easily over here, but it takes a lot of time in the human cell. Sir Alexander Fleming and his work in discovering the penicillin. And this is the last slide we are going to discuss, how it came to happen, this scientist to find such found that saved a lot of lives. He was working on bacteria and he left the plate. He came back later and found this big thing over here. Those who work in microbiology, they call this plate with this extraordinary thing, unusual thing over here, a contamination, because this plate must contain only the formal and semi-formal shape of colonies like that. Those are bacteria. So he found this weird thing, and then you can know that the more we go toward it, the less that we can find bacteria around it. So after analyzing it chemically and physically, it found that it's one of the fungi called Penicillium Notatum, and from the penicillium you can figure, yes, this is the main source of penicillin, which saved a lot of lives in the World War II.